like making stuff out of nanotechnology. Generally, this will be using DNA. Most of the teams in the past have used DNA, but it's not just limited to that. And what you choose to make, it can be something that's useful and could have a commercial application in the future, or it could be something that you think is really cool. Usually there's overlap between the two. The easiest way to sort of like show how what it's about is to talk about some examples. So one example is the team, the Dresden team, Dresden DNA. They came second last year in this competition, and they had a really cool project where they arranged DNA, where they used DNA to arrange gold nanoparticles along like a surface, and then that could be used to transmit electromagnetic waves, so making cool little mini circuits. So that's an interesting example of it. Another example would be the project of the team last year, which was based at the Victor Chang Cardiac Research Institute, and about half of them are here right now, Robbie, Lawrence, and John. For those of you who don't know, the competition takes place at Harvard, and these guys won the competition at Harvard. So to give a brief introduction to like what this is about, we'll just play the video. Hopefully sound works. I hope I didn't just crash. Oh, it's on internet, so that's why.
I do actually.
John's got it.
Yeah, I think it's working. It's just slow.
Yeah.
It's about 5,000.
5,000. And for those who are coming in now, we're just playing the video from the team last year.
If you ever wonder how an ant makes decisions, or how a cell knows when to divide, or how does this little bacteria decide whether to keep swimming or turn around, it's due to the fundamental principle of cooperativity. And we're the first group to use this principle to build better molecular beacons. A molecular beacon is a biosensor that is designed and built from a length of DNA. When it binds to a DNA signal, it unfolds and emits light. This type of beacon can help identify pathogenic organisms, viruses, even mutations. But because there are so many different noisy signals in the environment, the degree of sensitivity required can sometimes lead to false positives. So how does nature overcome problems like these? How have biological systems evolved to turn these complex environmental signals into simple binary outputs?
Observe these remarkable ants. The decision that the colony must make is whether or not to move to a new nest. This is, of course, a binary decision, but it's informed by many complex environmental signals. If ants acted non-cooperatively, each ant would make decisions independently of the other ants in the colony. This means that some ants may elect to leave while others would remain. But ants tend to behave cooperatively, meaning that each ant's decision affects its neighbors.
Do you guys smell that? What are you guys talking about?
The more ants decide they want to move, the more influence this will have over the rest of the colony. Ultimately, the ants will either stay or move as a group.
This cooperativity is based around the fact that the state of each subunit can affect the state of its neighboring subunits. This is a principle seen at all scales in nature, such as in the regulation of cell division, or in the proteins of the bacterial flagellum. In fact, it was this flagellum that provided our team with the inspiration for our design.
Our team has used DNA origami to construct a brand new kind of beacon. Individual molecular beacons are joined together in a ring and connected with linking bands, so that when one of them finds a signal, its neighbors are more likely to also find a signal. By adjusting the length of the linking bands and the signal threshold, it's possible to fine-tune both the cooperativity and sensitivity of the biosensor. In addition, we've developed mathematical models of these parameters, which allow us to feed back into the design and adapt the sensor to suit many applications with high sensitivity and low false positives. Coupled with an amplification system and a cheap, simple detection device, this could replace slower, more expensive detection systems in developing nations and health crises. By drawing inspiration from nature, our team has not only designed a novel biosensor with many potential applications, but our results may also shed some light on some of nature's fundamental principles.
Alright, so that was work of the team last year. I'm not going to go into too much more detail about exactly what. Oh god.
The next YouTube.
In the late 1960s, young.
The YouTube's still going on. Anyway, I'm not going to go into too much more detail about what DNA nanobiotechnology or any of that is. They'll take quite a lot of time. It's a very broad field and it can be used in many things. If you want to find out more, then there are examples on the internet, like the wikis of past BIOMOD teams, two of which we just showed. So the main point here is that this was all done by real undergrads. Undergrads who, before starting the project, didn't necessarily know like how you could make these things out of DNA. But by doing this project and practically applying this and actually making something, they kind of learned how to do it and developed some pretty schools in the process. The rest of the session will talk more about some practical things if you're interested in being part of a team. It'll briefly cover the history of the team so you know what's going on, what we might do in terms of a project and the likely timelines which will be involved, which is a key thing for anyone. We'll also cover what we might be looking for people to be in the team, future opportunities to work in this, and also some frequently asked questions that we've got. Afterwards, Robbie, Lawrence, and John. Robbie and John were members of last year's team, and Lawrence is the advisor who will also be advising this year's team. They'll talk about their experiences, and they'll either tell you how much they love it, or they'll tell you it's a trap and run away. Then we'll have Daniel from New Zealand Innovations talking about something, as well as conclusion from Winton, who isn't here. He's getting pizza in the rain. So if you, when he does do his little talking, make sure you give him a huge round of applause. So firstly, just on how the team sort of formed, my understanding of it is Robbie and Andy, someone else, they heard about BIOMOD and they attended the talk Lawrence went to, and they thought, this is cool, let's do this. And Lawrence was like, maybe. And then Andy was like, YOLO, and invested his own money to register the team. And and the team formed. They did work throughout last year. They attended the competition, and and they did all right. So that's a brief history of the team. Regarding what the project will be, this is about what we know so far. So the cool thing is that whoever becomes the team for this year, whoever is involved with this, they will have a really exciting opportunity to design whatever the project will be. There are some ideas kind of floating around about what it could be, but really, if someone in the team really wants to do something, and basically the project selection thing will probably look like 100 people will come up with about 100 ideas or so, and most of them will be pretty bad. A few of them will be okay, and then they'll be evaluated, and from that, one will be chosen. In particular, I think something that's really cool about this is DNA nanotechnology is kind of it seems to me as though it's a technology which hasn't really been applied very much so far. So if members of a good interdisciplinary team, which isn't just biology students, but you also have students from physics, electrical engineering, and computer science, if a team consists of like a bunch of different people with different interests and skills, then you could go about actually maybe making like a prototype of a device which tests something, like a physical device that you hold in your hands and can plug into an iPhone or something. So that's cool. Or we could just go like some completely different direction and make some self-assembling robot or something. So who knows? This is probably the most important bit, what the timelines are likely to be like. So next Tuesday, March 31st, is when applications to be part of a team, they close. We hope to to select the team within the next 10 days or so, kind of around by the end of the mid-semester break? Yeah, mid-semester break. April to May will probably be learning about like what DNA nanotechnology is, and then coming up with all these designs and maybe decide, like hopefully by that point deciding what it will be. And yeah, lots of brainstorming. By the end of May, the project we want it to be somewhat finalized with an experimental plan in place. June is exams, so nothing's going to happen then. July will be sort of crunch time for this because July is a period when we'll have like a solid month to just do lab work and really get a lot of the stuff out of the way. And it will be a pretty intense time. Like the team will likely be working on it almost every day. A lot of it, pretty much every team. Actually, yeah, every team. Every other team is from the Northern Hemisphere, so they have their summer holidays, like which is June, July, August. During that period, so they have 3 months to do all this stuff. We have months, so it's a bit crammed, which is why it will be a bit intense. And we want to get as much stuff done in the July break to avoid having to do things when when the uni semester is on. So August, September will hopefully just be wrapping up lab stuff, working on the wiki. So there's something I didn't outline is a large part of this competition is documenting your work in the form of like a wiki or a blog, and creating a YouTube video like the one you just saw, and communicating what you did in a very simple, easy to understand and good way. So working on a wiki in October. We'll probably hopefully just be finishing up on the wiki and the video. And at the end of October, like on October the 25th, the competition kind of ends. Like you're not allowed to work on your wiki anymore, so you pretty much just have to stop and chill. From October 31st to November the 1st, that's when the presentation at Harvard is. It's called the Jamboree, so all the teams from everywhere come and present their work then. Exams start from November the 6th, so this is just on StuVac, so keep that in mind. So the main sort of takeaway from this is it will be intense. Previous members of the team have kind of described it as kind of a mini honors, like a bunch of students doing a little mini honors over the winter break and afterwards. So, and you should be prepared, like we'll try to do everything in July, but probably be prepared to be doing a reasonable amount of work in August and September, and hopefully not October as well. So just something to keep in mind, like by the nature of like the people who are interested in this, a lot of people that are into this might be sort of the type A overachievers who like run a business, do 5 subjects of uni, run a charity, and are also like astrophysicists in their spare time. If you're one of those people and you think you'll be struggling with time, just think carefully about whether this is something you'll be able to commit to, because it would be pretty bad to so that's most of that. Regarding what we're looking for, in this year or this round of BIOMOD, like this entry of it, we're looking to have about 6 to 8 people in the team. That's like the team limit according to the rules, which is strange because I've looked at other team wikis which have like literally 20 people, but like you have to have the team limit is up to 8 people. So based on the number of people here, it's likely that there will be more applications than there are positions. So what we're looking for, which I'll cover in the next slide, is to make a really strong interdisciplinary team. So people with different skill sets in the team. It would be interesting if there was a team full of just like 8 mathematicians and no one who did biology. So we're trying to get like people with different abilities. Things that you can emphasize in your application would be technical ability. So like a science or engineering background. Biology is really important. Obviously, biochemistry is good. But like there are a lot of things that could be useful here, not just biology. If you have a strong background in like, it's kind of up to you to look at different projects and everything and see what skills were required in them and how you could fit in. Some of the projects in the past were very, very physics oriented. So something that uses, so a project which would use like a biosensor which uses lasers and different types of crystals to detect when something happens. That would require like very strong physics skills, people who are good at math, and someone who's good, maybe someone who's good at electrical engineering or mech engineering. So and emphasize where you think you could be useful. Skills like lab skills, there's going to be a lot of pipetting happening. Quantitative modeling, like math, I guess, is useful. Programming is really good. But there's also like a large aspect of the competition is non-technical. So like a big part of why the team did well last year was because the wiki looked amazing, the video was great, and that was because people like put in a lot of effort into making the website look good and all this graphic design stuff. So if you're good at any of those things, like the non-sciencey part, emphasize that as well. Like if you're really good at Photoshop and you do that stuff in your spare time, maybe like put a link into some stuff that you've done and say I'm a pro at sponsorship. And obviously like teamwork and communication are really important. If you can say that you've worked in like really stressful high pressure team situations, that's great. And yeah, this is an explanation of what just said. We're looking to form an interdisciplinary team. The project that we take on will be decided by the skill sets of the people in it. There's no like skill you have to have or like nothing like that. Just consider your strengths and how you could contribute. Another thing is this isn't just about like BIOMOD this year. This is about starting a sort of community, I guess starting like something for the long term. So we're not just looking for people to be part of the team this year, but also people with a sustained interest in this sort of stuff, like using DNA to do cool things or just working in this field and are interested in like devoting their spare time to like, developing skills in this area. So none of these are like definite. It's kind of one thing at a time, but we're hoping to do something like a lab fellows type thing, where people who maybe aren't like part of the team but are still interested in learning what we do and would be interested in being in the team in the future, they can come into our like lab in July and help out or watch us do lab work or something. So that will be a really good experience to like develop lab skills, see what's going on, and it will be fun. If you're interested in next year, starting a team next year, that will be cool. It would be great to have like more than one team happening next year. Obviously this year we can't do it because like we're starting off and there's funding requirements and everything. We're hoping, well, depending on interest, we'd like to run kind of like workshops or something or learning opportunities about this field. So yeah, keep that in mind as well. And the application, it's pretty straightforward. Email that email address with an application letter, CV, and transcript. The application letter is on our Facebook page. It just has 5 questions like, why do you want to be on the team? Why do you think you'd be like good for the team? But there's also a bit on what do you think you might make, and don't be too intimidated by that. Like just think about, just like survey the field a little and think about what you might want to do in a project or like cool things, cool areas where this sort of stuff could be applied. And there are some FAQs, frequently asked questions that have come up in emails. One is like, I don't know anything about DNA nanotechnology, is this okay? Not that many undergrads really do. I don't think I do either. I don't know about the team last year, but yeah, John says he had no idea before he started. So don't worry about that. You can pick up stuff on the fly, that's cool. The other thing is, can you apply as a first year? Yes, but be sure to like highlight how you can add value to the team. If like you don't know that much because you're in first year, then that's okay. But just make sure you clarify how you can apply. Can I apply as a non-biology student? Should be obvious now. The team will mainly work at UNSW. Probably, yeah. And the other question is like, will I be funded for going to Harvard and doing all of this? We're looking, we're hoping to get funding from like the government and the university and other grant schemes. So hopefully crossed, yes. And yeah, so that's the end from me. For the next 20 minutes or so, so now it's like the chance you have to ask all of your questions. Firstly, Robbie, Lawrence, and John, in that order, will talk for a bit, and then if you like, feel free to just bombard them with any question that you have. Like, really go for it.
Great, awesome.
Cool.
I hope you guys have any questions from that. Like, feel free to interrupt at any point if you have a question or if I'm not being clear. I think Sid did a great job of introducing the competition. Things that I add, like things I'm imagining the questions you have at the moment, so like getting your head around exactly what this competition is. The size of the competition, for instance, is like roughly 30 teams from all around the world. We were the first team from the Southern Hemisphere. The teams differ in size. I think the smallest one I saw was 3, and they include high school students working with a PhD student from New York, to like Sid was saying, 20 from the German team. And part of it, as Sid was saying, this field of DNA nanotechnology is kind of new, and it doesn't necessarily have an application yet. And that's the way I see the competition. That it's like, okay, here's this cool technology. It's like precision engineering on a molecular scale. What can we do with it? Let's like throw a bunch of undergraduates in a room, and it'll be an awesome educational experience for them, and they might come up with something awesome. Yep.
Sorry, I'm sure this has already been done before. Has anyone created a Turing machine out of DNA?
Yeah, people have started to make similar like cellular automata and stuff like that. And there's a whole literature on making DNA computers. Stuff like that. Yep.
Just to add to your question, if you check literature on DNA computing, you'll find lots of examples of calculations with DNA. It's really interesting. It's limited in some ways and so much better in other ways. So it's really cool to check that. So DNA computing, and have fun.
And there were a couple of cool teams who did stuff last year. One of the amazing things about going to Harvard is just the diversity of projects that were there, and also just meeting young, passionate scientists from all around the world. That was a really awesome experience. Other stuff I was thinking you might have questions about, like what does it involve in terms of time commitment. Sid mentioned this, and it would be awesome if you guys are that organized and you get most of your lab work done in July. I don't know how that's going to play out. I expect, like, our team started really at the start of semester 2, and then it was all co-curricular. Like, you're at uni and you have to timetable, when some people are on lectures, and then somebody's going to finish the experiment that afternoon. And I expect it will be like that for you guys as well. If you can get organized and be a great team, and that's awesome. But it's a big commitment, and you shouldn't go into it unaware that it might affect your grades. It might take a lot of time, like personal time that you expect to be at home on the weekend or something like that. But this is speaking from personal experience. It was totally worth it. Not just in terms of for itself, the experience of doing research and having a genuine experience of being a scientist. Because you have science lectures a lot where you learn scientific information. You have practical classes where you do scientific protocol. But when you go through the experience of designing a whole project from scratch, laying out all the experiments, doing that lab work, as well as stuff like finding funding, there was like team media stuff, and then trying to bring it all together in one concrete package like the wiki and the presentation you give at Harvard to be presenting before your peers. It's like the first genuine experience I had of being a scientist, and I think that in itself is worth more than all the courses that I've missed out on time from. So that decision is up to you, but you should be aware of what you're getting into. One of the things Sid talked about was how you choose a project. And it was awesome that he had like a blank slide, because you should definitely feel as a BIOMOD team you have scope to choose a project. However, it's also really, really scary and really hard to start with a blank scope. And for us last year, it was really helpful having Lawrence as a supervisor. And I'd just like to describe how we came up with a project, because when Andy and I started from scratch, we came up with these crazy ideas like the DNA Turing machine sort of stuff, and we wanted to make XNA origami, so we used like nucleic acid strands that aren't even DNA to have XNA, DNA, that sort of stuff. We're like, we'll do this. We had a bunch of other crazy ideas. It was awesome meeting with Lawrence, and for about 3 hours, we went through every project in the lab, and we learned how to get PhD students doing this, postdocs doing this, all these different projects, and then it sort of emerged out of that there's all these resources and all this expertise lined up around this question of cooperativity, and every person in the lab is working on that question in a different way. And at the end of it, it just seemed kind of obvious that, okay, here's the whole playground, the expertise and the resources, and we come up with the project out of that meeting saying, okay, what if we build an entirely cooperative system from the bottom up? It's a pizza, woo! Yeah.
I think last week was talking about how if you had like someone from a physical background who make advice to like a late advice to set some things. Is the project based around making DNA nanotechnology, but then there's scope for making something else to be used in tandem?
I'm not 100% sure I understand, but is it like just research or is it like just technology or something? Is that correct?
Is project you just revolving around DNA nanotechnology, or is it, can you make other things?
Yeah, there are definitely other things that are still at that sort of scale of nanotechnology. There were some teams who did sort of surface chemistry, some people did like membrane-based stuff, and you can look into this by looking up the other BIOMOD teams. However, most of the teams were DNA based. Yeah.
I'm sorry, to go to like carbon, like how long do you go for and how much approximately does it cost?
Yeah.
How much funding do you get and how much do you have to put in?
Right. Like last year we went over for something like 4 or 5 days. 2 of the students stayed over for longer time because they wanted to stay in New York after they finished their exams. But that was up to them, and they spent their own money over there. So you just kind of like, I think John was in the middle of StuBac, so he came back straight after for exams. So the time you go over depends on what you're doing. The cost is roughly around $2,000 for a return airfares per person. There's also the cost of sort of registration for the competition, which is about $250. Isn't much. Accommodation was subsidized last year by one of the sponsors of the whole competition. This year, I think there may be a small fee for teams, like a third of the price. So I'd say something like $2,500 per person to go. Hopefully, the team should be able to raise funding to support the teams, and there are a few opportunities like Sid mentioned. One is the New South Wales Chief Scientist has announced a grant round specifically for things like competitions like BIOMOD. There is also the opportunity of being supported by the university, and that would be, say, if you have students from the medical sciences and from the School of Physics and from engineering, you would approach the heads of school that they represent, and then maybe also the Dean of Science, and then maybe also the Vice Chancellor of Education. In that way, you can sort of leverage funding for groups in order to find enough to support all the team members. However, if the team isn't successful in doing that, yeah, the team members should be aware that there's a possibility that if they want to go, they might have to pay them first. Any other questions about that? Yeah. One of the other questions they had just before they were asking for it was about how the team actually works and how you imagine the lab work playing out, especially if it's an interdisciplinary team with all its kids from different backgrounds. It would be different for this year's team than last year's team, but I was going to talk about our experience last year in case it helps. We had weekly meetings every Wednesday at Arvo, and it would just be like a big catch-up session because the different people in the team sort of naturally started leading different parts of the project. John made an awesome video. He also did a lot of the DNA origami design stuff because John has a background in industrial design, so he knew 3D modeling of products, and he applied that to 3D design of DNA nanostructure. I did the lab work sort of stuff and also helped out a little bit with the model, which was actually headed up by Cyril, who's a physics and medicine student. But everyone on the team kind of gravitated towards a project, and they were leading that area. But it's not like you only make the wiki if you're good at computer science or programming or something. Everyone's involved with every part of the project, but usually back at the team meeting, you'll be catching up to, okay, this person's been doing this, so cool, this person's been doing this. Yeah, does that explain how the team works or not really? Cool. Do you have anything to add, John?
No, sounds good.
That sounds good. I'm going to move on. I think I've said it. Yeah.
What do you get for winning?
That's
a great question.
Well, it's just really small, like nanoscale. So we can't really show it off.
Really shiny certificates.
Yeah, we got some shiny certificates. That's the glory, I guess, of like we did this awesome thing. It's the feeling of you shared something amongst all your peers around the world. You do well. Just going there and listening to all the other students from around the world and presenting your own work is awesome just in itself. So yeah, what do we get for winning? But kudos. Yeah.
Yep. So I was just gonna add something to the answer. It's a bit of a holistic answer, but don't underestimate that the actual process experience is what you get. Because if you need something at the end to be motivated, you're not fit for the team, right? Because it's a lot of work. What you're gonna learn from that, I promise you, you're not going to forget. You're going to say it was worth going to uni for that in one of the lectures. You're going to say that.
Yeah.
If you're part of it.
That's interesting. The means is as valuable as the end. It's like if you're interested in this project, it's not because you go and win this thing at Harvard. You'd be interested in it because you'll learn something during the process.
If you do decent work, you're going to enjoy it and you're going to learn something from it. If you do a crappy work, then you're not going to enjoy it, of course. But if you do something decent, whether you win or not, it's going to be a great thing to do.
But yeah, whoever the team is, whoever you guys are, it's a great position to do well in the competition, to be competitive. There's going to be John and I around to sort of mentor the team this year, and Lawrence is an awesome supervisor. Any other stuff? All right, John, Lawrence, do you want to talk about your experience as a supervisor? What was that?
I mean, Sid and Robbie have kind of spoken about everything, so I don't, have a lot to add in terms of the introduction. I'll just continue on with that last question of what do you get from winning. It was bloody cool when they won. Like I couldn't go to this particular meeting, and I remember I woke up one morning and I didn't get much. They gave me 2 words: "We won." That was it. That was just that was awesome. So it is really cool, and teams do. Like the Dresden team, for example, I understand they're in every year and they came second every year, and this is the first time. Last year's was the first team, not just in Australia, but first team in Southern Hemisphere. No one had heard of the Victor Chang. There's no one in Australia doing DNA nanotech at all. We're the only lab, and no one knew that we were doing it. So suddenly, out of the blue, this team comes from Australia, and everyone's wondering, well, who the fuck are these people from Australia? And they took out the grand prize in Harvard. And it does generate serious kudos, because there are a lot of people that listen to that. Robbie's been contacted by certain people. I've been contacted by people who are interested in, even investing in us. What's this technology going to go? Where are we going to push it to? And we don't really know where it's going to go. So it is cool winning. But I'd say that it is true. That's not really what it's about. I mean, my motivation to be involved with this, initially introduced from Robbie and Andrew when I saw them present their iGEM project, which is another undergraduate competition run by MIT. My motivation was about education initially. So it's like when if you study philosophy, right, and you go through your philosophy course, but you never learn to think. Right? You just learn what all the other philosophers thought. And it's kind of similar with science and probably less with engineering, because you probably create stuff, but certainly with science, and I'm a scientist. You learn so much about the scientific process and what other people discovered, and you do predefined experiments where you get a fixed result. What you never get is the experience of discovery, and that is really the thrill of what we're doing here. Like, there's so few occupations, if any, where you are literally given that blank canvas which Sid gave you, and you could do anything you want, right? And there are 2 things which are so important about that. One is your freedom, and the other thing is that it's actually fun. If you're going into this experience because it's a really hard slog and I'm climbing uphill all the time, then it possibly isn't the type of experience for you. You need to be in this space where you really do have this freedom to say, oh, wow, I've got this blank canvas. I've got this technology where I, as easily as a professor in Harvard, could come up with the key innovative idea which could beat everyone else, or could be, blow the world away. And the truth is that each one of you has that capacity. There is no reason why. There's a Q&A, Q&A, the ABC show, and they had all these famous scientists on this Q&A panel, and a postdoc in my lab was in the audience at the time, and he asked them, I think it was Peter Doherty, one of the Nobel Prize-winning scientists, and he said, how can Australia compete against, the Chinese who are putting millions or hundreds of millions of dollars into science? How can they compete against Americans, blah, blah, blah? I think the key answer to that is innovation. Innovation is really how David can beat Goliath in this, because it is really true that in the right spirit, with the right team, in the right environment, you guys could come up with an idea. It's free, right? You could sit here now and come up with an idea. So my motivation is really to give you guys the environment and the space to say, okay, this is what my lab does. What we do is we study biological systems and we try to build them, right? And part of that motivation is to say, well, biology has existed for 4 billion years, so it solves a lot of problems. Can we build one of these things to learn how it works and maybe use these things to create technological devices like these physicists are interested in, right? That's our canvas. You know, we have so many different techniques, so many expertise in our lab. We have a fantastic group who are all willing and able to work with you. Our lab's actually moving to a new institute at UNSW, which should be the premier institute for single molecule fluorescence in Australia. And that's your blank canvas, right? And the key that I'd like to bring out of you guys and the mentors I'd like to bring out of you guys is that spark of innovation. And that's really, if you guys can come out with that experience and then in terms of the legacy, you pay that forward. I mean, it's kind of infectious. It started when I saw Robbie and Andrew, and they were just kind of like frothing at the mouth about research, you know, and you really rarely see undergraduates like that because you don't get that experience. And I promise you, if you go into this and you have the right motivation, that you're going to come out the same way. And that's really amazing. And it doesn't even matter if you go into science. It doesn't matter what field you go into. If you can maintain the aspect of freedom and fun in what you do, then wherever you go, you're going to have a great legacy, I think, in your life. So that's really my motivation for keeping you guys, or having to, you know, running the BIOMOD competition in my lab. Are there any questions from the supervision perspective or from the lab perspective or the kind of things we do in terms of research? Yeah.
Just with time management as a whole.
Yeah.
That enthusiasm that you talk about, is that like an overarching enthusiasm that you have when you look back on, or is it something that every day when you meet or do the project and interact with it, like you're like excited about it and, or I'm doing this, I'm getting away from university, or I'm doing this, I'm excited to like progress this, or is it kind of something you look back on and you say that was really great?
I don't know. The team can answer independently. From my perspective, it's always exciting. Like I come to work and like John and Robbie and Andrew are now in my lab, and I really look forward to, like, John and I have meetings every couple of weeks, and I really look forward to our next meeting because John's often come up with this really cool idea. Oh, that's awesome, you know, and that's always exciting and fun to me. There are obviously aspects where you have to document stuff, which is kind of less exciting, but it's like any kind of job. But I'd say far and away the dominant feeling is excitement throughout the process. That's at least my perspective. And that's not just with BIOMOD, that's just with science. Like, because you really are on that blank canvas. It's like you're swimming in a space which no one's been there before. It's like you're in a little rowboat rowing on a completely blank ocean, and you're stumbling across these beautiful islands. That's kind of, I would say, the modus operandi of, you know, discovery. The team might have a different perspective. They might say, "Oh, I totally sucked," and Robbie and John might want to add to that.
Yeah, I would say it's a mix of things. I totally agree with everything Dr. said. I think that was an awesome speech. Impromptu action about discovery and the nature of the scientific process. But yeah, there are parts of it where it's like you're doing an experiment. You have to do the same thing for an hour or something to set up an experiment in order to get a result. But yeah, there's the excitement about the anticipation of is it going to work or is it not, and then it doesn't, and it's actually just more confusing than, okay, it didn't just not work or it didn't just clearly work. It's somewhere in between, then you have to puzzle through that, and it's fun in itself. So, yep.
That element's like I think there are two things that are different to, I mean, if you're just filing paperwork, for example, there is two things which are different to what Robbie's saying when you're doing that extra experiment. Yes, the pipetting can be like tedious, but you've put together that experiment, you've designed that, and you have a stake in what's going to come out of that. The other thing about that is that when it doesn't work out, it's not much different to, like, you know, people for fun, they do like fucking Sudoku puzzles and crosswords and all these kind of things. They do it for fun because solving puzzles is actually fun. The difference is this is a puzzle which you could actually have a result which actually has an impact on the world. So it is slightly different to just the tedium, I think, even when you're doing those tedious experiments.
I thought it was a pretty interesting question. So I'm just gonna say that it's actually both, you know. But about the overarching, experience, it's something that you can only feel after you have a few years of science behind you, right? If you're still young, you cannot look back and, enough things to get a feel of what to achieve. So I mean, when you're younger, I think the main aspect of the fun is, every day doing something that challenges you, and you get high on it. Really, it's really exciting, and it's something that you want to do more and more, I think, and so on. But once you've done this for a number of years, you look back, and it's a different sensation. You look back and you look at it, and you go like, oh shit, I did something myself. I did something that matters. But this comes later, right? When you're still exploring this, it's mostly, the everyday thrill, I think. But if you stick to science, you get to experience both, which is, a more complete experience, hopefully positive experience. So you find both. I mean, something like biomod, you do your work every day, hopefully motivated. Once you've done it, then you can look back and you see the whole picture, and that's a different thing. So you get both of it.
Any other questions? Yeah.
You mentioned like impacting the world with the project. So what sort of whole world impact do you reckon was the result of last year's project?
Right. So that was kind of based on there's so many on that. So one thing which I think everyone understands is this whole thing about sensing, right? And what I was saying earlier is that in nature, because it's had 4 billion years of evolution, so many of the problems we have, like sensing, has actually been solved in nature. This cooperative switch was based on how a ultra-sensitive switch in nature actually works, right? The problem is we thought we knew how it works, and generally this is the way biology works. So I'm going to start again, because that was a really garbled response. Okay. The whole concept in this particular project, and I'm going to get away from the sensor because it's not really what it's about. The concept of this particular project was whether or not you can build biology. And the reason why that's important is because biologists aren't like engineers, or they're not like physicists. They're not actually used to building things, right? And there was a famous quote from Rutherford who said all science is either stamp collecting or it's physics, right? And what he's basically saying is physics was actually deriving fundamental laws that it could explain the entire universe. What are biologists doing? We're looking at a plant and poking it and saying, oh, it's green. Or you're tearing it apart and just trying to see how it works. And actually, biology hasn't changed so much. The reason is it's so complicated that we were not in a position to build biology. It's basically it's what's called a top-down approach. A biologist would, rather than try to build you, they would take some of your skin cells and you'd poke it and prod it and try to work out how it's responding, so work out its work. Or you take a car and lift up the bonnet, right, and look inside the car and see how it works, right? What did this last biomod team do? They took a completely fundamentally different approach to biology. They said, this system, we think we understand enough to actually try to construct it from scratch, right? Why is that important? The difference is if you're going to open up a bonnet of a car and work out how it works, you could look inside the car and see that, oh, there's a fan belt, and every single time that car's on, that's spinning, right? And this is the way biology works, right? You generate this kind of model which correlates the way the car works. And you think, okay, it must be that spinning fan that drives the way the car moves, right? And that's a perfectly fine correlative model, right? What you'll never discover is that it's not a rotating fan that spins rotating wheels. It's actually a piston that moves up and down, but you'll never ever know that. What they did is they said, we'll just give them all the parts and we're going to try to build this from scratch. Once you can do this, then you can take nature's designs and do whatever you like. One example of this is to create an ultra-sensitive biosensor. And actually, this created a lot of publicity during the project. We had Channel 7, Channel 2. We actually had a riff with Channel 7 because we said, okay, we're going to build a new sensor. What can we sense? Let's do Ebola, right? Because it's kind of cool and we have a hit for the media. But the fact that it caused a spat with media was simply because we did Ebola and then they put our photo up with a dead patient, right? And that was just way too far, taking the concept way too far. But what was actually true about that is that sensors like this can actually create new technologies to detect things like Ebola and HIV and different viral diseases. So that's one kind of impact of this project. That's a long-winded, ramble answer. I don't know if that answers the question.
I reckon that there's actually a class coming here at 6 o'clock.
Oh.
So what we're going to try and do, if we got pizza or something outside, is we can keep answering questions and stuff like that. We chat outside. John, Lawrence and I will be here. If you guys have any questions, come up and chat amongst yourselves. If you have questions about the application process and stuff like that, let us know. Yes. Sorry, yeah, there's something you wanted to say.
Yeah, it's fine. Good team, like really cool for them. And then you could actually sometimes that develop and so on. It's good for something. It's fine. Good for me. So I'll just like actually something and send it to the base there. Anyway, thanks a lot for